So all bacterial cells are surrounded by a cell wall made out of peptidoglycan. In the laboratory, one of the first things we do to identify a species of bacteria is we examine just how thick their cell wall is. We do a procedure called gram staining, and we determine the gram status, as we call it, of an organism and divide that big, huge group of bacteria into the first distinction, which is whether it is gram positive or gram negative. And this is all based on how much peptidoglycan is found in their cell walls. So if an organism is gram positive in the laboratory, what that means is that after we do the gram staining procedure and we look under the microscope, the cell is going to stain purple. And what it means is that that cell has a very thick layer of peptidoglycan in its cell wall. A bacterium that stains a pink color with gram staining is called gram negative. And that organism, that cell, has a very thin layer of peptidoglycan in its cell wall. So we do gram staining and we determine what we call the gram status of a bacterium because it turns out that it's the first way that we can classify that bacterium by the anatomy of the peptidoglycan in its cell wall. Is it a thick layer or a thin layer? The thick layered organisms are the gram-positive type, and the thin layered organisms are the gram-negative type. Now, I want to mention that there is one type of bacterial organism that you can't gram stain. Even though you apply the stains and you do the procedure, when you look under the microscope, the cells are neither pink nor purple, or I should say, they don't take up the color particularly well. We refer to these as being gram indeterminate. Their gram status is indeterminate. The other way we describe these cells is acid fast. And we use that term, acid fast, because we're saying that the only thing we can stain these cells with is an acid stain. Now, the reason that this particular bacterium doesn't stain with the gram stains is because it has a layer of wax around its cell wall. Now, this particular type of wax is called mycolic acid. There's a picture down at the bottom. There's an image that was taken under the microscope. These are not individual cells of this organism. This is what we call a colony of the organism. It's growing on an agar plate, a petri dish, and there are actually thousands and thousands of cells growing in each one of these mounds that you can see on the agar plate. What I want you to notice is that they have sort of a waxy look to them. They look like the kind of wax that you might pull out of your ear, for example, or other types of sort of natural waxy material that you might see. This is mycolic acid. And again, these cells, this particular organism, secretes mycolic acid around its cell wall. So when we try to stain them with gram stain, we can't do it. They just don't pick up any color. This is a very important feature of this type of organism. It helps us identify them. And the organism I'm talking about is called Mycobacteria tuberculosis. So its genus is Mycobacteria and its species is tuberculosis. And yes, this is the organism that causes the disease that we call tuberculosis. On this slide, we can see a very simplistic drawing of the difference between a gram-positive bacterial cell and a gram-negative bacterial cell. And remember, these designations are part of how we identify bacteria. We talk about their gram status. So what you're seeing in this turquoise blue color, this is the peptidoglycan layer, and it's sitting outside of this structure, which is the cell membrane. So we can't see the entire cell here. We're looking at a little piece of the membrane around the cell. So the whole cell would take up more than this slide. The whole cell would extend way beyond the limits of this slide. We're just looking at a little piece of the cell membrane around the cell. Remember, it's a bilayer. You can see the heads and the tails in this membrane. You can see a protein in this dark color. So this is just an ordinary cell membrane. And this would be the inside, the cytoplasm of the cell, and the outside would be over here. And in the outside area, we see this thick layer of peptidoglycan. So this would be a gram-positive bacterium. Now compare that to this drawing over here. Here's the plasma membrane, the cell membrane. Here's the inside of the cell. Here's the outside of the cell. Here's a thin layer of peptidoglycan. And then outside of that, there's actually a second membrane. Yes, a second membrane. This is a gram-negative cell. It has a thin layer of peptidoglycan compared to this gram-positive cell. And outside of that thin layer, there is a second outer membrane. These are the types of structures we see in a gram-negative bacterial cell. So gram-negative cells are quite unique. In gram-negative cells, there's a cell wall made out of a thin layer of peptidoglycan and a second membrane. That second membrane is very similar to the plasma membrane in that it's made out of phospholipids and it's made out of protein, but there's a special molecule that comes off the surface of the outer membrane that I want us to be familiar with. This is a molecule called lipopolysaccharide, and we shorthand that as LPS. LPS is also called endotoxin. Now, if you translate the word endotoxin, it means the toxin within. Endotoxin is just part of the cell for a gram-negative bacterium. It's just part of its structure. But when you and I have a lot of gram-negative bacteria inside us, and we all do, that gram-negative bacteria can become dangerous to us under certain circumstances. Now, again, the LPS molecule is just part of the structure of gram-negative cells. In fact, it's there because it provides some protection to them. It actually helps to strengthen that outer membrane. That's why the bacteria have LPS. And normally the LPS doesn't bother us. But when a large number of gram-negative bacteria all die at the same time inside of the intestine, and all of those gram-negative cells break up and fall apart, the LPS molecule becomes very dangerous. This endotoxin gets released from the cell, and it can cause us to go into shock. In fact, it can even kill us. Now I'm saying us, meaning humans, but endotoxin can harm any animal. that goes through some kind of a medical crisis involving the gastrointestinal system, where large numbers of gram-negative bacteria all die at the same time. So you can imagine perhaps there's a twist in the intestine and a blockage. Perhaps the blood supply to the intestine gets compromised. Perhaps there's a terrific trauma to the abdomen and to the intestine. These are the kinds of things that sometimes happen and can lead to the death of large numbers of gram-negative bacteria that normally live down in our gut. And when a lot of those cells die at the same time, they release the LPS molecule, and it just so happens that it's quite toxic to us. And it causes, again, a shock to occur, and even sometimes death. So here's another drawing that represents a piece of a gram-negative bacterium. This represents the inside of the cell over here, and this would be the outside over here. So here's the cell membrane with its bilayer of heads and tails. Here are some proteins in purple sitting in the cell membrane. Here is a thin layer of peptidoglycan shown in this light sort of teal color. And then out here, this is that outer membrane. Again, it looks like an ordinary cell membrane. What's unique about it is that there are these molecules sort of extending off of that outer membrane. This is LPS, or again, what we call endotoxin in medicine. Now, the next thing I want to talk about involves the plasma membrane of prokaryotic cells. So just the ordinary cell membrane of prokaryotic cells. This membrane does something that the membrane in eukaryotic organisms doesn't do. So this is unique to prokaryotes. The plasma membrane, the cell membrane around a prokaryotic cell, is where that cell makes its ATP for energy. This is really interesting. A eukaryotic organism, like a human, has organelles in its cells, remember? And one of those organelles is called mitochondria. And inside the mitochondria is where ATP gets made during a metabolic process called cellular respiration. Prokaryotic cells don't have organelles, so prokaryotic cells don't have mitochondria. They still need ATP, they need energy, so they make their ATP using their plasma membrane instead of their mitochondrial membrane. So the way this is done, and we're going to talk about this more when we talk about microbial metabolism, but the way this is done is that the cell is going to build up a gradient of hydrogen ions, so H plus ions. And that gradient is going to be built up across the cell membrane. So there's going to be a difference in the concentration of H plus ions, hydrogen ions, on the inside of the cell versus the outside of the cell. And we're going to give this concentration gradient a special name. We call it the proton motive force. That word proton here just means hydrogen ion. The proton motive force. Remember, nature doesn't like gradients. Nature likes to get rid of concentration gradients, and this gradient is no exception. So, what a bacterial or an archaeal cell is going to do is take hydrogen ions, protons, and transport them from the cytoplasm across the plasma membrane and into a little space, a little cavity in between the cell membrane and the cell wall. So in between the membrane that surrounds the cell and the peptidoglycan layer. There's a little space, and we call this the periplasmic space. It's just a cavity in between the membrane and the cell wall. The prokaryotic cell is going to send out hydrogen ions into that periplasmic space. It's going to actually pump them out into the periplasmic space and build up a gradient. So there'll be lots of hydrogen ions outside the cell in that periplasmic space, and there'll be many fewer hydrogen ions inside the cell in the cytoplasm. We've got a gradient. Now remember, one of the things we said about simple diffusion, in terms of the things that can use it and the things that can't, we said any molecule that has a charge on it cannot cross the membrane using simple diffusion. Well, a hydrogen ion has a positive charge on it, so it can't cross the membrane by simply squishing in between the phospholipids. Instead, we need facilitated diffusion to get rid of this gradient. And what's going to facilitate it is a special protein called ATP synthase. ATP synthase. This is a protein that is actually an enzyme. And if you're not familiar with what an enzyme is, it's what we call a catalytic protein. It's a protein that's capable of helping a chemical reaction occur. So this protein, ATP synthase, like the name suggests, is going to help in the chemical reaction that builds ATP, that synthesizes ATP. And this protein is going to help get rid of that hydrogen ion gradient. The hydrogen ions are going to flow through this protein and into the cell. Remember, we said when it comes to facilitated diffusion, some of the proteins are channels and some of the proteins are carriers. And we said channel proteins actually have a tunnel or a hole right through the center of the protein that the molecules can travel through. And that's what ATP synthase has. ATP synthase sits in the cell membrane, and it actually creates a tunnel. And the hydrogen ions actually flow through that tunnel and back into the cell. That gets rid of the gradient. That makes nature happy. It gets rid of the gradient, and it generates ATP in the process. That's how prokaryotic cells make their ATP. On this slide, we can see a very simple drawing to show us where this hydrogen ion gradient builds up when a prokaryotic cell makes its ATP. So we're looking at what represents a prokaryotic cell. Here's the inside of the cell. Here's the cytoplasm. The DNA would be in here. The ribosomes would be in here. The red line represents the cell membrane or the plasma membrane. And then we can see this blue line out here. This represents the cell wall. And out here would be the extracellular space. So remember, we said in between the cell membrane and the cell wall, there's a little space. And we call that the periplasmic space. And when a prokaryotic cell makes ATP, what it's going to do is it's going to send hydrogen ions from the cytoplasm across the membrane and into the periplasmic space. It actually pumps those hydrogen ions out. So we're going to build up a big gradient. There's going to be lots of hydrogen ion here, not very much in here. We've got a gradient. And the way the gradient resolves, the way the gradient goes away, is by having the hydrogen ions flow back through the membrane into the cytoplasm. through a special protein, and that's what we're looking at in this light green color. This is ATP synthase. It's a protein, it's an enzyme, and it's sitting in that red cell membrane. The hydrogen ions are going to flow through the tunnel created by the ATP synthase back into the cytoplasm, and in the process, ATP will be generated.